Welcome to Homework Lib.
Hello students, for the given question, first part, the two generalized phenotypes associated with tasting the chemical PTC are tasters and non-tasters. Tasters perceive PTC as bitter, while non-tasters do not perceive any taste or only perceive mild bitterness. Second part, PTC and the TAS2R38 gene are special because they play a significant role in the perception of bitter taste in humans. While other taste receptors have broad ligand specificities, TAS2R38 is specifically tuned to detect PTC and related bitter compounds. Additionally, variations in the TAS2R38 gene can result in different sensitivities to PTC and other bitter compounds, leading to differences in taste perception. Third part, the TAS2R38 taste receptor is a G protein coupled receptor, that is GPCR, that is expressed on the surface of taste receptors cells in the tongue. When activated by PTC or other bitter compounds, TAS2R38 initiates a signaling cascade that ultimately leads to the perception of bitterness. Fourth part, SNP, that is single nucleotide polymorphism, is a variation in a single nucleotide at a specific position in the DNA sequence of a gene. SNPs can occur naturally and can result in different versions of the same gene known as alleles. Fifth part, the names PAV and AVI come from the three SNPs that differentiate the most common tasted allele PAV from the most common non-tasted allele AVI of the TAS2R38 gene. The PAV allele has 3 amino acid changes proline, alanine and valine, while the AVI allele has 2 amino acid changes which is alanine and valine. Sixth part, based on the location of the polymorphism in the final protein, it is more likely that the AVI protein or AVI form of the protein is effective in binding rather than signaling. The polymorphisms in the AVI allele result in changes in the binding pocket of the TAS2R38 receptor, which may affect its ability to bind to PTC and other beta compounds. Seventh part. If you know the phenotype for PTC tasting, you can make the following hypothesis about the genotype. Tasters have at least one copy of PAV allele which encodes a TAS2R38 receptor that is sensitive to PTC and other beta compounds. Tasters may also have two copies of the PAV allele or one copy of the P allele and one copy of the AVI allele, which can result in different levels of sensitivity and other beta compounds. Non-tasters have two copies of the AVI allele which encodes a TAS2R38 receptor that is not sensitive to PTC and other beta compounds. Non-tasters may also have one copy of AVI allele and one copy of non-functional allele, which would result in non-tasting. I hope you find the video useful. Thank you.